a partnership project led by the Prince William Sound Science Center. Our partners include the UAF Alaska Sea Grant Extension Office, the Cordova chapter of the Prince William Sound Audubon Society, and the Forest Service, who donates the space and equipment. My name is Meadow. If anyone has any ideas for lectures or subjects in the future, please let me know. And I'm going to be passing the sign-in sheet around, so make sure you put your name down so we can keep track of attendance. And next week we're going to have Clay Coughlin from CEC is going to be talking about the potential Crater Lake Power and Water Project. So that should be pretty interesting. And tonight we have Scott Pegau from the Science Center, who's going to be talking about the various hearing research and where things are at right now. Scott. Yay, Scott.
Okay. So, as you can tell, my name is buried inside this because I'm actually presenting results basically of a lot of other people's work. And so I'm just here to try to give you an overview. This program has been funded by the Exxon Valdez Oilspill Trustee Council. And we are currently in the fourth year, closing in on the fifth year of a 5-year research program associated with hearing. So just a quick outline of the talk. We're going to talk about the existing program, the one that we're in. Like I say, we're in year 4. I'll give you an update on results. These are just kind of highlights from the many projects. And then quickly kind of take a look at where we anticipate going in the future, although we are early enough in the process that we can actually adjust the program at this point. So that's part of presenting it now. It's a great time to get feedback on it. So with the current program, the goals was to improve predictive models of herring stocks through observation and research. We broke it out and we said, okay, we'll try to do 4 different things to address that. One is provide information to improve the age-structured analysis model, and we're going to do it in a couple ways. There was a required synthesis effort, so it was, okay, let's pull all the information that we have and try to, tell what the status of our knowledge is. There's a lot of measurements that have been going on over the last 25 years. And some of them have some pretty big assumptions that had never been tested. And so we said, okay, let's stop and let's do some testing on these so that we know that we're doing the measurements right. And we're also looking at new approaches to monitoring and kind of understanding hearing. The design, if you've been around for a while, you've heard a lot of different programs. So Sound Ecosystem Assessment was a EVOS funded program that occurred in the mid to late 90s. And it looked at pink salmon and herring. Then there was, EVOS funded a lot of individual herring projects, and then in about 2008 they decided, we're tired of having all these one-off projects. We want to do integrated programs. So they came up with the Prince William Sound Herring Survey Program, which ran from 2010 to 2013, which took all the different herring research projects that were going on, put them together so that they would have people talking a lot better. The program that we're talking about tonight, I call it Herring Research and Monitoring, or HRM, that is focused in Prince William Sound. I take enough grief from my friends from when I lived in Homer that why aren't we covering Cook Inlet? And it's, well, I have a hard enough time covering the work that needs to get done in Prince William Sound, so we're going to focus here. It's a mix of monitoring and process studies. Monitoring is counting things, process is trying to understand why things happen. And we're dependent on the Gulf Watch Alaska program. And Gulf Watch Alaska is the long-term monitoring program that Exxon Valdez Oilspill Trustee Council is funding. And so that's looking at the oceanography, it's looking at the birds, it's looking at the whales, so it's looking at everything on either side of the fish. So when we want to understand herring in context of the ocean conditions, we need their information. One of the other things that we try to do is be responsive to local needs or information. A couple of years ago, one of the local fishermen volunteered to go out to Kayak Island to pick up a sample if we'd pay for the gas. And we needed fish from out there for genetics work and other stuff, so it was a good way to be able to work with the community to actually collect fish from an area that we normally wouldn't be able to get them. So what this is all the projects that we have going on over this 5-year period. And not all of these are 5-year long projects. Some start and stop throughout. And then I've kind of broke them out. There's by which are the objectives, and in the blue ones you have both monitoring and process studies. And I just want to kind of show you what the size of this program is at this point, and when we hit the future you'll see what's, how it's going to be changing. So what I want to do now is just kind of go through each of those projects and pull out one highlight of them. So disease surveys. They're doing regular surveys. They're looking at 3 diseases. I'm a physical oceanographer. I always use this as my excuse. I cannot pronounce words over 3 syllables long. So ichthyophonus I can say because I practice a lot, but I don't go into VHS or VEN because they are too long for me. But basically, ichthyophonus is one of the diseases that's pretty prevalent. A fish can survive it without a problem. It's a question of when does it become lethal. And we have a prevalence of about 30%. It changes year to year, and that's pretty typical for stocks around here. VHS, which is believed to be the one that probably took down the population in the 90s. It's a really nasty viral disease that kills fish in a real hurry. Over the last 7 years, I think they found 1 fish out of several hundred that had it. So our prevalence is running at 0. VEN is basically the fish goes anemic, and they're not finding that here either. Spawning, the adult biomass surveys, all I can say is spawning was strange in 2015, although it was strange in 2014, and it was strange the year before that. I could swear that there were 3 years where it was predictable, but that was probably just because I didn't understand it. But basically, there's a lot of small spawn. There was spawn on Montague Island, a good amount of spawn on Montague Island, but basically it came down to, we had a hard time getting a good acoustic survey. Our number was fairly low, where the Science Center survey was in the kind of 12 to 16,000 tons, and the estimated population the year before was up around 22, 20, and so we did not find a lot of fish, but we may have been out too early because the spawn occurred for quite a while after we surveyed. Juvenile Index, Pete Rand is taking over this project. He's a new researcher at the Science Center, and so I don't have an up-to-date result because he's getting his feet underneath him and trying to understand the data he has. Age 0 condition, most years what we find is that the age 0s, those, little fish, 3 to 4 inches long, by February, March, those fish are starving, and there have been a couple of exceptions, and when we get the exceptions, those appear to be really good years for recruitment. Aerial surveys, 2013 and 2015, we had large numbers of age 1 herring observed when we flew in June. And this 2013 flight at age 1 is a 2012 brood year, and that was one of the few years where the age 0 fish came through the winter in really good shape. So they went into the winter with a high energy, and they came out, and that appear to translate into large numbers of age 1s. Some of the process studies, one of the things that we're trying to do is determine the age of first spawn. It's a project. There is a maturity function in the age-structured analysis model that helps try to figure out, since the sampling is the spawning population, you want to know the total population, you need to know how many aren't spawning in a given year. And so trying to determine what the actual number is, it's an interesting one because on my advisory groups, one says, oh, you don't need to do that. We know they're all recruited by age 3. And the other one says, oh, you don't need to do it. We know that only a few of the age 3s have recruited, and you can use the age 3s to figure out everything else. And so when they talked to each other, they kind of said, yes, this is an important thing, because even though we both think you don't need to do it, we can't agree on what the answer is. What we have found is that sometimes we can see a bimodal structure in their growth, and it's assumed that if you are going to spawn, you grow slower than a fish that's not going to spawn, because you have to put energy into eggs. And so the idea when we first went in was to look for this bimodal growth to figure out how the population was splitting between ones that were going to spawn and which ones aren't. The problem is we just aren't getting enough years where it's a clear distinction, and so we're going to probably have to go back at this in a different fashion. Genetic stock structure. Now, this was, for me, probably the most interesting result that I've heard in the last 3 years, and it's a preliminary one. You know, this could easily change, but the preliminary result is that when they looked at the genetics of Yakutat, Kayak Island, and Prince William Sound, based on the markers they looked at, they were the same fish. But when they looked at Cook Inlet and Kodiak, those were extremely different fish from Prince William Sound and different from each other, which really shocked me because I know those two populations mix through the winter. You know, and so for me it's always been, yeah, I can throw something in the water. It takes 10 days to get to Cook Inlet. You know, I've thrown enough drifters in the water to see them hit Cook Inlet in 10 days. But we are not exporting juvenile larvae to Cook Inlet. And they're in that planktonic stage for over 3 weeks. They have plenty of time to make it. But genetically, it's, you know, as she was trying to explain it to me, she was saying it's about as different as the Copper River and the Ski Lac. You know, if you were looking at salmon, they're just totally distinct populations. We also have gone back into the age-structured analysis model that Fish and Game runs and rebuilt it in a Bayesian framework. It's a statistical framework that allows us to take a look at the probabilities of a population being above a threshold. And we've been going through it and taking a look at the inputs, the data that we have, and we're pulling one out at a time and seeing how well the model performs to figure out which ones are the most important. And this is, down here is a model run, obviously 2013, showing the estimated population and the probability of it being above the lower threshold. We had a synthesis that was required of the program. Here again, what EVOS is looking for is for us to get all this information that's being collected and put it together and try to make something greater out of it. You know, it's data is knowledge, but understanding is when you gather all the knowledge and try to pull it together. So they're trying to get to an understanding. And part of what we wanted to do was take a look at some of the historic data that we haven't had a chance. And so Steve was actually doing a scale analysis from the how many hundred thousands of scales do you got in that?
About 300,000.
300,000 scales. You know, so able to pull back fish all the way into the 80s. There's scales into the 70s, but you know, just trying to process them. And one of the things that we saw is this red dotted line was about the minimum size in the first year that they had grown. And so if a fish was caught out there in a spawning population, it had grown this much in its first year. Well, we looked at the size of fish that we were catching, these age 0 fish, to figure out what that meant. And basically, a fish has to be over about 85 millimeters if it's likely to survive. And it turns out that number shows up again elsewhere, that when the fish are going into the winter, these age 0 fish, they have this competition between, they want to grow length and they need to store fat. And so what they end up doing is they grow for length, they put their energy into length until they hit about 85 millimeters. And then you can see them make a shift from growth into adding lipids, to putting on fat. And so it seems to be a pretty critical kind of size for a herring to be able to make it to live through the winter. Addressing some of the assumptions in our measurements. One of our assumptions was that, or we had a model that suggested that once a herring went into the winter, and we were thinking, okay, winter for a herring starts in about November, that they were going to stop feeding, and once they stopped feeding, they'd lose essentially a certain number of calories every day. And so that their energy would have this kind of linear decline. And what we found was, yeah, it peaked in November. They were able to put on quite a bit the year that we looked, between October and November, but they lost everything within a month. And so by December, they had lost all this gain that they had from October to November. They had lost it already. And by January, they were down at this whole body energy content of about 3.2, which is about as low as we see them. And we figure that's basically starving. That, you know, if you go below that, you're likely to die pretty quick. You know, it's not the energy, but it's the fat. And there is a lipid level that corresponds to that. That, you know, once you go below that lipid level, the fish are then metabolizing their heart and kills them pretty quick. We were sampling November and March because we were thinking that was start of winter, and March was the end of winter. Those seem, you know, there's a slight recovery in March, but that seems to be pretty good. And, you know, you really need, for a fish, it needs to make it to April to be able to, you know, coming out of March and into April that they start really feeding. Having said that, I keep saying start really feeding. One of the things that we did find is those fish are feeding through the winter. And so what we did was look at the fats that the fish have and use different types of fats. And if a fish was truly starving, it would basically it was expected that both of those fats would decline at the same rate and you would stay as a group. And so you wouldn't really move on this chart. And what we did find is that there is movement, which is suggestive of them feeding through the winter. You know, and when we look at stomachs in March, we are finding that they are feeding, but they're not metabolizing enough to prevent that energy loss. And some places are better than others. You know, fish generally that are on the west side tend to start off fatter and have less of the signal of eating through the winter than like Simpson Bay. Simpson Bay, those fish, they come in smaller than the west side, and they tend to have to eat a lot more to make it through the winter. One of the other things that we're doing, that juvenile index cruise, we go out and we survey 8 different bays in Prince William Sound looking for how many age 0 herring we got, because we want to use that to predict what the future future recruitment might look like. And so the question was, okay, when I go survey each bay one night, how representative is that? And so what this is, if you just look at dark blue, basically these are 1, 2, and 3 are all 2 weeks apart, or it's 2 weeks between 1 and 2 and 2 weeks between 2 and 3. And what we see is, depending on what week we went out there, we could get a 10-fold change in the estimated density of fish that we saw. Which means that we have to really rethink how do we survey these juvenile fish. And we're going to, and where that becomes really important is when you want to try to estimate survival, because if you're estimating survival, you have to have a really good estimate of how many fish are there at the start and how many are there at the end. And right now, we're not able to capture that kind of quality of data. Direct capture is really to validate the acoustics. It's its main purpose, but it has shown some interesting results. One of the things that we thought was happening going into this was that the young fish like to hide under ice. And so, on a normal, really cold winter, you'd have those ice shelves form in the different bays. And in this winter, this survey is from Simpson Bay, and there was a big ice shelf in there. And when the ice shelf was there, this is the size of fish they caught. And so this, around 100 millimeters, that's the age 0s. This is the age 1s. Here's the age 2s. But when the ice went away, and it went away the last day of a 3-day cruise where they were sampling the same place for 3 days in a row, and when it went out, and they went in and looked, now all of a sudden most of the fish that they found were age 0 fish with some age 1s. And basically what it was all the age 0s were hiding under the ice where they couldn't trawl. And so the juvenile fish, this is one of the signs that yes, the juvenile fish really are hiding under the ice, most likely to avoid predators. Disease studies. One of the things that came out of the disease is this ichthyophonus, and we report it in prevalence like I did it early on, where I said, okay, about 30%. Well, what ends up happening is about 5%, a little bit more, each year probably gets ichthyophonus. And so if you're 10 years old, you've had 10 times to be part of that 5%. And so you have a much higher chance that you have had that disease somewhere along the line. And so long as your body is working well, you're going to actually just isolate the ichthyophonus, and it's going to be present through the rest of your life. So you can catch it as an age 0 fish and have it as an age 12 fish.
How old do they get?
We got measured up to age 14.
Yeah.
Yeah. And so, it's, so the thing is when you report prevalence, this is what we're suggesting is that you have to actually report your age structure. Because what this is showing is the age structure, or this is actually length structure, where you've got your largest fish is yellow. And this year the prevalence was low, but that was because there were no large fish caught. And so it becomes really important to be able to say, was this year a low prevalence year, or did I just have a different size structure of the fish sample? And so that's changing how we'll probably be reporting ichthyophonus prevalence. Some of the developing new approaches, here's where a lot of the fun stuff tends to happen. Tagging, and I don't know if you've heard Mary Ann's presentation, where basically put acoustic tags in adult herring, release them, and where you see these arrays, these are essentially acoustic receivers. And so as the fish swims across, it's got a pinger in its body that tells the receiver, this fish crossed here on this day. And there is actually some up here. Well, of this, 43 to 69 fish were observed down at the entrance. We had 3 fish that stayed in Gravina for 2 to almost 3 months. There was still a fish being detected when we pulled the arrays that were in that area. But most of them screamed out of here after they had surgery. So it took 2 days to get to Hinchinbrook entrance and 3 days to get to Montague Strait for a fish after it completed spawning. And so those fish were able to move in a hurry. The fish stayed down here. Some of the ones that went to Hinchinbrook entrance showed up at Montague Strait, and a couple of them that started at Montague Strait showed up at Hinchinbrook entrance a little bit later on. But they stayed down here till about July. So after they spawned, they came and they hung out down here. In July they disappeared, and then in September they reappeared. Unfortunately, this array is a single line, so you can only tell the fish has crossed it, and you don't know which side the fish left. You know, it can come in and then turn around and come back. So we don't know if the fish actually went out of the sound or not. We definitely suspect that they did. And so that's something that we want to take a look at in the future.
So your range doesn't go any further?
No, it just goes across, and so you know you need to have 2 receivers so that you can tell which direction the fish is actually going, and our array wasn't set up that way. Disease forecasting. So one of the things that we think we really need to do is not necessarily worry about the prevalence. You know, that tells you if a disease is out there, but what is the likelihood that you will get that disease? So we want to do a couple things. We need new tools to monitor these. So VHS, if a fish gets VHS, 90% chance it's going to die within, I think, the first 10 days. If it survives, it's golden. It will never catch VHS again. And it doesn't have any markers in it that you can look at a blood work and go, okay, this fish has had VHS. The only way to know that is to try to give it VHS and see if it lives. So been working on new tools to actually determine if a fish has had VHS in the past. Because if all the fish out here have been exposed to VHS at some point and they're refractory, then you can't have a big disease outbreak. If all of these fish out here are naive and have never seen VHS, then you can get a rampant outbreak of these diseases under certain conditions, and that's what we want to know is what's the potential for that kind of outbreak. We've also been looking at the transmission of ick. It was 4 years ago. The only way they were transmitting it was to actually inject the fish. They've figured out that you can actually grind up infected fish and then feed it to the herring, and that will allow them to get the ichthyophonus. That actually, unfortunately, came from taking a look at fish in the harbor. The little juvenile herring in the harbor have a very high prevalence of ichthyophonus compared to outside the harbor, and we're trying to guess, okay, what might be the causes, and one of them was, okay, the ground offal. Particularly halibut tends to have a lot of ichthyophonus, and so is the potential, and so he tried grinding up infected fish and it worked. We don't know that that's why it's happening in the harbor, you know, it was, but that was from looking at the harbor, we were kind of going, okay, let's try this. And we learned how to actually give fish the disease in a little bit more natural way. Non-lethal sampling, so we're out there catching fish. I like catching fish. I like chasing fish. But sometimes it's a question of can you do it without actually killing the fish. And so what we did was use the remotely operated vehicle with the cameras, and then there's a Ditson sonar underneath. And we'd use it to find schools of fish. And then we were actually able to use the Ditson to measure the length of these fish that are on the order of about 10 millimeters, and run in with a camera and identify them. And it turned out, we also used this under the ice to confirm that the juveniles were under there. And one of the things with the little age 0 fish that was just kind of fun, and I probably should have put the movie in on this, is that when we went at night, they were attracted to the lights on the ROV. And so there was one where we go down, we find this big school, and then we slowly back out, and the school follows. And you can see all these fish staring at the camera because it's got the lights, and they're all kind of following as it comes up. Which is not really good for making measurements of them, but it's really good for identification because they come right up to the camera and you can see exactly what you got. The last kind of big component is coordination, synthesis, and outreach. Kind of goes over all of the different projects. We turned in a synthesis. It was actually the second one in less than 2 years that was provided to ELOS. If you're interested, the first one really looks at age 0 herring, what we know about them. The second one expands out a little bit more, takes a look at all the results that we've been getting from the different groups and putting them together. So, the fact that, the fish that were spawning had all reached this minimum size, that also showed up in the food. And, it was pulling together all these different projects, and how they actually tie together is the second one. We try to work with ADF&G as much as possible. We work with the Golf Watch program, we work with CDFU, we're trying to do our best to coordinate outside. There's a website, and also obviously education efforts, and we've drafted Fish and Game people in the past in those efforts, obviously. But a large part of the synthesis is trying to make sense of what graphs like this were telling us. And basically, really what this one tells us is that the further up you are, the fatter you are as far as the fish. It's the whole body energy density. And then the further over this side is actually the carbon isotopes in your body, whether it's depleted in carbon-13 or not. And we think that this is a sign of food from the Gulf of Alaska versus food that was produced in Prince William Sound. And so generally, if you are more negative, you tend to be a fatter, more fit fish. And then if you look at the colors, the ones that tend to be on the right-hand side of this is Simpson Bay, and the ones that tend to be on the left-hand side are Whale Bay or Eagle or someplace on the west. Taking a look at how does this connect to the oceanography, what we have plotted, one of these days, so get the right button. This is a plot of the first year growth, so how large the fish grew in a given year versus the average of the July and August temperature. So basically in July the fish is metamorphoses, it sets its scale, and it seems that the warmer it is in July and August, the faster the fish is able to grow it. Except for 3 years that were exceptions, 1989, 2004, and 2005, which were all very warm in the ocean, but had poor growth. it's okay, yeah, maybe temperature has something to do with growth because it's going to change the metabolic rate. But you would really think it's associated with food. And the food records are not nearly as good as far as the length. But one of the things that we looked at is we used actually data from out in the Gulf of Alaska from the continuous plankton recorder. So this recorder is towed behind a cargo ship that runs from Long Beach into Cook Inlet. And we looked at the portion on the shelf. And this top one is the diatom abundance anomaly. So diatoms are one of the types of phytoplankton. They have an essential fatty acid that has been shown to be important for herring growth. And that is the best relationship that we have. So this is the last 10 years, which includes 2004 and 5 in here. And we get a very clear relationship between the diatom abundance and the growth of the herring. We don't know if it's because of the quality of the food. We know, like I say, that diatoms have an essential fatty acid, or if the fish are actually consuming the diatoms when they are young, and that's really helping grow. There's some evidence that they do consume phytoplankton when they're small as well. When we look at it to zooplankton, there is a relationship, but it's not nearly as good. And we were thinking going in that the relationship was going to be with zooplankton because that's the primary food. But it's not associated with zooplankton abundance. It may be a certain type, but we've looked at large copepods, small copepods, several different others, and it's never as good as it was with diatoms. Keeping an eye on predation. This is from a student's thesis. It's the estimated whale population in Prince William Sound up till 2009. I put this blue line in for 1989 because quite often you hear, well, the whale population has doubled in the last 25 years. And based on her estimates, it was somewhere around 100 whales in 1989, and most of the time we're probably up in the 120, 130 whales in more recent years. And so not seeing that doubling in the whale population, even though it's well known that the whale population in the North Gulf of, or not North Gulf of Alaska, but North Pacific Ocean has doubled in that time frame. It's not clear that we've had a local doubling in the number of whales. Humpback whales, yes. They've changed the survey approach since 2009. I don't have comparable numbers. And a lot of it's trying to figure out, the whales are, most of them are transiting through. So they come in, they feed, they leave. And a new one comes in, feeds, and leaves. So it's really trying to figure out how many whale days the fish are exposed to is something that trying to get teased out. This is Rich Brenner's photo. I flipped it upside down. I like it just because this is the herring eating the whale. So finally got a chance to get even. First time I saw it, I saw the Loch Ness Monster eating the whale. And this is a school just outside Titlik, where the whale was cruising up the beach. So looking at the future, well, the EVO's Trustee Council released their request for proposals for the next 5 years. They're saying, okay, yes, we'd like to do herring research again for the next 5 years. Effectively, it's about $250,000 a year smaller. The total number of dollars is actually increased, but we had an overlap with a previous program. We've managed to get several things added on to the existing program, and they kind of scaled us back to an original formula. So we're going to have less money coming in. They state as a goal that they want to continue the development, testing, and update age-structured assessment model in collaboration with Fish and Game. So they're following fairly close to what we had been using as as our goal for the last 5 years. And then they list several areas of interest. They wanted, how do the changes in birds and fish affect predation? They asked if we would hire a postdoc student that might address some of these different questions, trying to get some youth into the program. As for comparative analysis of stocks, which we actually have underway, we're already doing. Comprehensive spawn assessments, make sure that we're getting good assessments. Adult herring movement, so following up on the tagging. They were really interested in what we were able to get from the tagging and looking at that. They really like the disease work and want to continue with that to try to figure out, what's the potential of actually having an outbreak. Relate herring recruitment or growth to oceanographic conditions. I showed you one example, but they were looking for expanded. What's the tide whale predation? This age at maturity is returned that yes, they are still interested in trying to figure out that age at maturity, because it's a function that's just really hard to get good data to do. Effects of climate change, and then anthropogenic changes. And this is really, there's two that are hidden in that word, and that's the hatcheries and the oil spill. We aren't going to be able to address all of those, but those are what EVOS identified as areas that they would like to get proposals to contain. Additional factors, as far as I can tell, the ADF&G is not going to have a budget for herring monitoring. If there's no monitoring, I seriously doubt that there's going to be a push to open a fishery. And here I am looking at most of the Fish and Game employees and happen to say this, but I think that that's the reality of it. And that ADF&G is going to need to collect some of that data to be able to go. So when we look at the future program, one of the important components is going to be to ensure that that data remains collected so that we know the status of the fishery, or the fish stock. I shouldn't call it a fishery. We know the status of the fish stock, because without that, we have, that is the thing that we've been trying to build a lot of our research off of, and without it, we're not going to be able to say anything about what's happening to the fish out there. The Trustee Council in FY16, so this coming fiscal year, approved funding to purchase tagging equipment. So basically they put a big investment into tagging before the next 5-year program started. So kind of taking that as a hint that the next program probably should have a good tagging project associated with it. And the trustee, staff, and science panel have been promoting the postdoc. They keep looking at me and saying, "You're too old. You need someone younger working there." And then they specify how much money and how often they can work and stuff like that. And we go, "Oh, that person's not full time. How am I going to attract a younger person if I don't have a full-time job?" But they really would like to see that. So what do I think that the program is likely to look like in the next 5 years? We'll have components that are associated with the modeling. So continue work on the population modeling. I know they use a structured analysis, and at some point I would hope that we could start adding in more of what I call first principles, more information that's based off the fish than just the statistics. So that's why I call it population modeling. Take a look at this. Look at that, determining the age at first spawn. So this is kind of in the model. These are inputs to the model. So the adult biomass surveys, I'm hoping that we'll be able to keep the aerial juvenile surveys. I'd like to be able to keep, one of the big predictions was that this year we'd have a large recruitment event, and you don't have the samples to be able to say whether or not the age, how big that age 3 population is. But I did get word that Kodiak had a very large age 3 recruitment event this year, and Sitka had a very large age 3 recruitment. So I'm pretty sure that we got the large recruitment. We just weren't able to get enough samples to be able to show that. I expect that we will work through EVOS to keep funding for the age, sex, length, and spawn surveys, aerial spawn surveys done by Fish and Game, because those are, if without those, we're not going to be able to say a whole lot about the fish. Other work will be the disease studies, herring tagging project, and then there will be coordination outreach and potentially post-op. But we are at this stage that we haven't put a word on paper yet, and we can actually adjust things. So if you have suggestions, now's a good time for them. Thank you for your attention. Yeah.
Who is Golf Watch?
Golf Watch, it was originally called Long-Term Monitoring, and it just wasn't a fun enough title, so they called themselves Golf Watch. But it's Exxon Valdez oil spill trustees decided that they wanted to fund 2 large programs, essentially for 20 years until their money was spent out. And they said we were going to spend it on herring and on ecosystem monitoring. And so that ecosystem monitoring is Golf Watch. And so they look at oceanography. So like Rob Campbell, his stuff is part of Golf Watch. The continuous plankton recorder. There's oceanography with the University of Alaska Fairbanks and the Seward Line and in Cook Inlet. Killer whales, humpback whales, seabirds, actually forage fish. They're trying to find all the other forage fish rather than herring is a component. And so basically either side of herring, they're working, trying to monitor how the ocean is changing. So the idea of if we can follow it, we might be able to tell the difference, or we can recognize what natural variability is. So if there's, hopefully we'd be able to pull out an anthropogenic effect like an oil spill.
They have a really dynamic website, tons of information in there. They pretty well got it easy enough to searchable stuff. It's worth a look.
Is that the fun you think?
No,
Something
no,
you're
everything's individual for me, but the whale trips,
the
the
whales
whales were part of the
Yeah.
Golf Watch. I was about ready to say the same thing. They do a really good
Good
website.
ones.
Lots of information.
So is as a part of your proposal, for the spawn surveys, that's that, but so part of that is the aerial surveys.
Yep.
So that is part of your proposal, or is that?
So the way this gets built is I go, Steve, would you write a proposal that includes this component? And then I will go to Paul Hirschberger and say, would you write a disease component? And Trevor Branch, and ask him, and would you write this component? And Kristen Gorman, and so basically what I'm looking at is what are the components, and then I go try to find the right person to put together a proposal to do that work. And so I'm looking at what EVOS has said, this is what we'd like to see done, and then I look at the budget, and it's okay, what can I manage to fit in here? Right now, the way I've drawn it up, the thing that I probably regret the most in that is that I don't have the small, kind of high-risk projects, that the genetics, which, is providing probably some of the most interesting results. Although it wasn't what, they had originally proposed to look at just the genetics within Prince William Sound, and the exciting part is when they were able to look outside and connect that. That made it really exciting. The tagging project, which we did in this past 5 years, was just, can we do it? You know, can we put transmitters inside herring, release them, and have them survive? And, you know, of the 69 fish, we actually detected 66 afterwards. So, you know, there are only 3, and we didn't get the array into Gravina until after some of the fish had already been released. And so we may have missed some of the fish just because they took off so fast. And it was 2 days from surgery to Hinchinbrook entrance. So, you know, they'd hold them on the boat for close to a day, I think, and then release them as a batch. And those fish, no problems. They were able to take a big transmitter and swim away.
How are you gonna explain that?
Add receivers above and below the lines so that we can see, we can say for sure, is this fish going in or out? What we'd like to do is tag fish that are spawning at different locations. So do Gravina fish go somewhere different than Fildago fish or Montague Island fish? See if there's differences there. Also want to take a look at if there's differences with size. We kind of think that younger fish are more likely to stay in Prince William Sound rather than leave into the Gulf of Alaska. And so want to test that to determine if, you know, that, you know, it's primarily older fish that are headed out and the younger fish tend to stay.
So were the aerials for the whales, which they're still at around 150? Do you have anything recent? Because this year was crazy for whales.
Yeah, you know, it's, I don't have, and it's, I have numbers, I think, to last year, but they changed how they're surveying. So the surveys I showed you, I'm pretty sure were summertime surveys based off of photographs, and now they've gone to doing fall, winter, spring kind of time period. And so it's how do I connect the new data with the old data is one of the questions that we really have. You know, and it's the whales. I know that when they went out in the spring, you were, I can ask Dave.
Yeah, that was one of the last ones, so
Yeah.
that is the drop of the program.
But it was pretty quiet, wasn't it?
Yeah, compared to the other years, what you say about crazy whales, that, you know, there's no counts or anything on the delta, you know, and offshore. It's only what comes into the sound that they look at. So
Yeah.
that's a whole nother world.
Yeah.
So I had a couple of questions. One, what really scared me as a Prince William Sound herring permit holder
Yeah.
is that they won't be able to, fishing game won't be able to tell if there's actually enough biomass to open a fishery, right? I mean, is that what you're?
If we are going off of the state's budget, that would be my guess, that they're gonna cut that program out because there isn't a fishery.
But that would be like the aerial surveys, and that how you judge it? I mean, is that, or?
No, it's just one component that goes into the egg structure model, so
Yeah.
it's got to
So it's like.
Yeah, you know, and that's why we're working hard to, you know, it's okay if we have to fold that component into the research, you know, we have to have that information. You know, all the other programs are really dependent on having an estimate of the population out there. And so we've got to keep, you know, components going so that we can get that on estimate.
Right. And one thing that kind of relates semi to that, now you're saying that the Yakutat and kayak and this stock are genetically the same, and that was some kind of people's gut feeling, oh, maybe
Yeah.
those ones were.
Yeah, you know, and the markers that they check, and geneticists, boy, they start backpedaling as fast as possible because it's this based on everything that we've looked at, there's no difference. But that doesn't mean that we've looked at everything, you know, but it's a lot of markers, it's 26 plus markers that they're doing, and, you know, they haven't completed all of the samples, you know, so they haven't done like this year's work and stuff like that. There's, you know, there's definitely more to come, but the early results were that these were similar and that was very different.
Yeah, that seems really important information and new information.
Yeah.
It really, of course, now you have to figure out what the whole biomass of all that is, but it seems a more bigger. You've got these, and there's no fishery in Habitat, there's no fishery out there, and
Yeah.
maybe we could have one here, but, you know, that's, you know, hopefully that can be followed up, because that really does seem to be a whole different perspective.
Yeah.
A whole paradigm shift, like, oh, this is not just our little thing here, there's
Yeah.
different conditions, so that one seems really important.
You know, and how do they mix, how do they exchange? Because I would, walking in October, if you had asked, in fact, early November, before I had heard the results, I would have told you that the fish in Cook Inlet were gonna look similar to Prince William Sound, because I just know how fast water moves. You know, it's half of the larval period, you know, it's easy to toss something out and get it there. But genetically, they're different. So for some reason, you know, there's not this larval flux that's moving fish down the coast. So what's happening that prevents those fish from surviving to reach that stage? And, you know, how are Kayak Island and Prince William Sound fish, you know, actually related as far as, you know, when they go out in the summer, do some of them just decide to stay out and they end up over at Kayak Island, you know, and that helps explain some of the fluctuation in the population level. Is it's actually changing? We don't know. You know, there's a lot to be learned about how those two populations relate.
I mean, I had the honor of watching them actually hatch. Oh, they lay their eggs, and then I was doing a study, and watching them hatch, and actually all of a sudden we're doing these eggs, looking at these eggs, and then all of a sudden they all are like little swimming. You can actually see them. You know, swimming around, and so, I mean, that's not a larval stage, right? They're like
That's what I would consider larval, because they're that until they metamorphose, and when they metamorphose is when they get scales.
They seem to have like a direction. They were like going and almost moving together. They weren't just drifting. They seemed to be.
They shouldn't be able to overcome the currents, but what they can do is go vertically up and down to catch different currents, so you can actually stay in one spot by just going up and down and catching the different currents that are at different levels. You know, there's limited, you know, they can swim to some degree, but, you know, you're not going to swim upstream against the current. Is basically what plankton is considered. Yes?
Is there any genetic information available on that Cook Inlet stock pre-Prince William Sound crash?
No. You know, I think that they were. Do you remember if someone tried to get genetics off the scales? Because that's the only.
It was the early 90s, but it may have been a year after the crash.
Okay.
The last time it was done.
Okay. Yeah, the Cook Inlet crashed at about the same time.
But if we wanted to do that, we could probably extract DNA from existing scale archives and Cook Inlet and Prince William Sound and compare them.
Yeah. Did you have? Okay. I thought I saw your hand moving around.
Thanks so much,
Fidgeting.
Scott.